Here we give some of the details of hemoglobin in the form of question and answers, with the idea that it would be easy to understand. What is hemoglobin? Hemoglobin is an oxygen carrying metallic protein molecule. Why it is called metallic protein? There are two components as the name indicates, Haem and globin. Haem, the main component of the Haem is iron. So because of the presence of iron with the globin, it is called hemoglobin. Now any non-protein substance attaching to the protein for structural purpose or functional purpose, it may be called prosthetic group of the protein. Accordingly, here the iron is the prosthetic group of the globin in hemoglobin. What is the structure of Haem called? Now the iron in Haem is present in the form of a porphyrin ring. So the porphyrin ring within which there the iron present is called a Haem unit. Structure of hemoglobin in relation to conformation of peptide chains is called, that is, in the case of hemoglobin, based on the structure of protein, that is globin part, it is called tertiary structure. Usually the protein is differentiated according to the arrangement of the polypeptide chain, appearance of polypeptide chain. You may be knowing the basic component of the protein is, basic unit of the protein is amino acid. So the amino acids join each other to form a peptide, which will be called a protein. And many amino acids join, that chain is called polypeptide chain. Even a simple understanding, if two amino acids join, dipeptide, three amino acids join, tripeptide, that way. Now if the polypeptide chain is straight in appearance, what is called conformation, it is called primary structure. And if the chain is in the form of a single fold, secondary structure. And if the chain makes folds in the form of a globe for the purpose of accommodation as well as function, this is called a tertiary structure. So in that sense, here the hemoglobin assumes tertiary structure. Where is hemoglobin? So hemoglobin present in RBC of blood. RBC, that you may be knowing, red blood corpuscles. So the RBC is a pack of hemoglobin molecules inside. There are millions of RBCs in blood, and every RBC contains millions of hemoglobin molecules in it. What is myoglobin and where is it? When we talk about hemoglobin, it's better knowing about myoglobin, a related word earlier. Now the myoglobin is also structurally similar to that of hemoglobin, that is made of iron and protein. But its location is very restricted, present only in muscles. Even there is a view particularly in skeletal muscles and cardiac muscles. Now if you say hemoglobin is to transport oxygen from lung region, that is alveolar region, to different parts of different tissues of the body. Myoglobin is its main function is to store oxygen, not to transport, present in myocytes, that is the muscle cells, so that can supply the storage of oxygen whenever there is a need because of the heavy and continuous work of the muscles. How many heme units present in one hemoglobin molecule? So as we told, one hemoglobin molecule contains heme and globin. There are 4 units of heme and 4 chains of globin with one hemoglobin molecule. Now, even if you see the next question, it can be easily explained. What are the contents of globin? Now, the globin is the protein part, we told, made of 4 polypeptide chains, and 2 chains are in a helix form and 2 chains are in a pleated sheet form. Accordingly, they are called alpha chains and beta chains. So, out of 4 polypeptide chains, 2 are in alpha form and 2 are in beta form. And the alpha chains are made of 141 amino acids each. Beta chains are made of 146 amino acids each. Now, these polypeptide chains appears in a form of globe, that is spheroid. Each contains one heme unit. So, one hemoglobin molecule in total will be containing 4 heme units in 4 chains, as each in one chain. That is one heme with one chain. So, there are 4 polypeptide chains with 4 heme units. Even here we can say one heme is able to attach to 1 molecule of oxygen. Accordingly, one hemoglobin molecule can combine with 4 molecules of oxygen. Why is the blood red in color? Now the heme and globin combines through different linkages, which makes the hemoglobin as a red pigment. So the expression of which in the form of the blood looks red, as it contains much amount of hemoglobin. What is external respiration? When we talk about respiration, there come 2 important terminologies: external respiration and internal respiration. External respiration is the stage of the passage of oxygen from alveolar region to the tissues, between alveolar region to a particular tissue. The oxygen is passing through the hemoglobin. That part of the passage of oxygen is called external respiration. Now, after reaching the tissues, what happens? The oxygen will get into the cells, cytoplasm. Tissue nothing but the collection of cells. So, within the cell, there the oxygen will be utilized for the formation of energy, which is called internal respiration. The usage of oxygen within the cell is called internal respiration or cellular respiration. So that way we can differentiate the respiration with the 2 words: external respiration, internal respiration. That is, the transport of oxygen through the hemoglobin is external respiration, and the usage of oxygen within the cell is internal or cellular respiration. How many hemoglobin molecules present in one RBC? As we told already, millions of RBCs present in blood, and each RBC contains millions of hemoglobin molecules. So here an approximate value ranging from 250 to 300 million molecules of hemoglobin in one RBC. What is the amount of hemoglobin present in human blood? Now it can be expressed in terms of gram per 100 ml. An average of 14.5 gram of hemoglobin present in 100 ml of blood. Even here there is a view in human relatively, in female human it is less compared to that of the quantity present in male. Anyway, we can tell it in terms of average. So 14.5 gram in 100 ml of blood. How much oxygen is carried by 100 ml of blood through hemoglobin? So 1 gram of hemoglobin able to combine with 1.34 ml of oxygen. And already we told the quantity of hemoglobin present in 100 ml, 14.5 gram. So multiplying this to 14.5 into 1.34 gives the value 19.45 ml of oxygen per 100 ml of blood. What is oxyhemoglobin? So the oxygen combines with the hemoglobin, forms a complex. It is purely a loose complex, reversible complex. And this reversible complex is called oxyhemoglobin. Even we have given it in the form of equation. Hb plus O2, HbO2, oxyhemoglobin. When it goes near the tissue, then the hemoglobin and oxygen will dissociate, separate, and oxygen will be entering the cells. What is carboxyhemoglobin? If the hemoglobin happens to combine with the carbon monoxide, it becomes an irreversible component, a strong component, and it is called the carboxyhemoglobin. What is oxygen dissociation curve in relation to hemoglobin? Now, when we say oxygen associates with the hemoglobin to form oxyhemoglobin at the alveolar region, that is the lung, and when it reaches the tissue region, there the hemoglobin dissociates, that is releases oxygen. So the oxygen tension or the partial pressure of oxygen at alveolar region, alveolar air, may differ from the oxygen tension after releasing the oxygen in the venous blood, and accordingly it can be recorded in the form of a curve, what is called oxygen dissociation curve. And here we give it in terms of value. 108 mm Hg as the partial oxygen pressure at alveolar region. And then after releasing oxygen at a tissue, then the RBC will return again to the lung region through the venous blood. So in the venous blood, partial oxygen pressure will definitely be less compared to that of the arterial partial oxygen pressure. So in venous blood, the oxygen pressure is 40 to 50 mm Hg. So it can be expressed in the form of a graphical curve, what is called oxygen dissociation curve, that gives the difference difference in the partial oxygen pressure between the arterial blood and venous blood. What is the lifespan of RBC? Usually once RBC produced, it works in a cyclic way. That is, takes in oxygen and transport it, release the oxygen at tissue region, again comes to the lung, takes in oxygen. That way it goes on circulating. But it becomes old. That is, you can call it a spent RBC in about 120 days. And such old RBCs are disintegrated at a spleen and a part of liver, and the Haem and globin components are separated, and then new RBCs develop utilizing nutritional iron, protein and all these in the bone marrow, and blood is refilled with new RBCs. So we are planning to give different topics. Even there are many more details related to hemoglobin. So we give it in the form of question and answer. We are planning to give it continuously. So subscribe, even make arrangement for the notification. It will be useful to you when you prepare for your exams. So subscribe.